This video will take you through the steps of editing the layout of a predefined assay protocol button. The BCA protein assay will be used as an example. You will also see how your BMG microplate reader can be used to perform incubation at a specific temperature. The BCA assay requires a 30-minute incubation at 37 degrees Celsius. The nano can be used to perform this incubation if you turn on temperature regulation as shown here. The target temperature has been reached. We can now turn our attention to sample preparation. Sample prep for the BCA assay involves mixing two separate reagents to create a working solution, and of course plating blanks, standards, and other samples, followed by addition of the working solution. After a lid is placed on the plate, it can be placed in the nano for shaking and subsequent incubation. While the plate incubates, we can work on the changes to the layout. Right-click on the assay button and select Edit Protocol. The type of plate can be changed, but mostly we're going to be concerned with changing the layout. After emptying the layout, the location of replicate standards and blanks can be identified. There are 4 replicates for each standard in columns 1 through 11, and blanks in column 12. Then the associated concentration for each standard can be included. In this example, the starting concentration is 2 milligrams per milliliter, and a 2-fold dilution series was used, so a dilution factor of 0.5 can be entered. Click on the header of the concentration column to fill in these values, and then click OK to save for later. The 30-minute incubation is complete, but the samples are supposed to cool to room temperature, so we will move the plate out. We still want to know what the temperature inside the reader is, so we will choose monitoring without incubation. Once the samples have cooled, remove the lid and move the plate into the nano for reading. Now you can simply click on the BCA assay tile and the assay will begin to run. You can monitor the assay's progress using the current state, where you can see the spectral data being collected for each well in less than 1 second per well. You'll also notice that as the protein concentration increases, the peak at 562 increases, which is what we expect in the BCA assay. Once the plate is read, you must close the current state to save the data in Mars, and then data analysis can be performed. Click Open Last Test Run to open this test in Mars. The BCA assay button is linked with a data analysis template, so some calculations have already been performed. The view we are currently seeing is a baseline correction for the data. Specifically, the OD value at 562 with a subtraction of the OD value at 750. Since we have already changed the layout, it will be very easy to use the standard curve wizard to create a suitable standard curve. Click on the wizard, check that all the settings that you want are appropriate, and click OK. The standard curve will appear once the calculations are complete. For this dataset, we can add error bars since we are working with replicates. And we can also check out the fit result to make sure that the R-squared value is suitably close to 1. If you have any questions regarding using predefined protocols or other nano functions, please contact us.